I just had a quick question
Okay,
about
yeah, sure.
the exam. Did you say it was going to be open 2 hours a day for 4 days?
No, no, no. It's going to be open for like 4 days.
Oh, okay.
Yeah,
Okay. I can't. There was a class that I had. The instructor said it's going to be open 2 hours a day for four
I
days.
got you. Yeah, yeah, yeah. No, no, no. I'm not doing that. Your exam is, I forget if it's, I think it's next week. It would make sense because that's week 4. And for what, yeah. My
Okay.
other class is on week 5, and I didn't make the schedule,
Okay.
but
Sounds good.
yeah, is the exam. There'll be a homework assignment. It's already posted in mastering. And I don't know if any of you had that problem on Sunday night when you went to go submit and it said it's late and gave you a 0. How many of you ran into that? Yeah. The rest of you turned it in a bit earlier, but sorry about that. I have no idea what happened with mastering. It wasn't even I looked to see if like I set the time uh zone wrong on it. Nope. Wasn't me. I have but if it ever happens again, for those of you that don't know, anybody that turned in the assignment after like 10:30 on Sunday night, learn said not learn mastering said it was 1:30. After 10 o'clock, it said it was 1:30 and basically assigned it late and then give you credit. If that ever happens, those of you, if you emailed me, you did the exact right thing. You showed me a screenshot that showed the time on your computer and the problem with mastering. And it took me a couple minutes to realize what was going on. Mastering didn't know what time it was. If that ever happens to you, don't sweat it. Shoot me a screenshot. We can fix it. Okay? As long as you completed the assignment, you'll get credit for it. Okay? And if you haven't done it, uh, go ahead and at least get it done so you can get some credit for it. You don't want to lose out on those 30 points if you didn't finish the assignment. But I don't know that anybody in here did not finish it because like 95% of the class, 164 out of 171 people completed the assignment on time. Good job. I'm impressed. And the average score was an 88%, 89%. So, man, that's almost everybody's making A's and B's. And then this week is up and it's all from the test bank. Same format. No time limit, so you're not being timed. It's just due on Sunday night. Okay. Don't sweat it if you're running late. Do it. Okay. Don't think just because 11:59 Sunday night has passed that you can't turn in the assignment for credit. Okay. Life gets in the way sometimes. It doesn't need it. We don't need to penalize you twice for life getting in the way. All right. So today we're going to finish up histology, the study of tissues here. And we've gone through most of them. And what we're going to do today is muscles and nervous system or nervous tissue. And we're just going to basically like introduce muscles and nervous tissue. And let me see if I can get that ceremony. There's like all these options here, like discussion panel, conference, ceremony, presentation. I just start pushing them until it looks okay. Just choose the one I like. Okay, getting back to this. So we're going to talk about muscle tissues and nervous tissue. And this is an introduction because we're going to spend a lot more time on muscle and nervous tissue. And I like talking about these two together, even though they're structurally really different. They're both what we call an excitable cell. Think about excitable, they're really exciting. Not just kidding. They are really exciting. What they do is like a muscle cell obviously changes shape when it receives a signal. So it can have a rapid response to a chemical signal. And then your nervous tissue, the nerve cells, they send signals down their axons and they need some type of stimulus to cause them to send a signal, and that signal is the nerve impulse, which is an electrical current going down the nerve cells. It's really cool how they do that. And to me it's always interesting like how fast they can do it. Like I watched NFL, right, and watching these athletes, like how fast they can respond to the environment is crazy. I mean, there's nothing like watching Tom Brady throw a 40-yard bomb and it lands right here in the guy's arms, right? Like, I could do that if I wanted to. Okay, so let's start with muscle tissue. And of course, muscle cells, or these are also called myocytes, M-Y-O. Myo means like muscle. And of course, you see the C-Y-T-E, site, that of course means cell. So these are excitable. They can respond to both electrical and chemical stimulation. Okay, so you're familiar, walking, breathing, heart beating. These are done by muscle tissues here. Muscle tissues can be really long, like the length of your hamstring. So we often call them a filament, a myofilament. Once again, there's that word myo. It basically means muscle. And then we have, in your myofilaments, there's striated muscle and smooth muscle. And then surrounding that is the endomysium, okay? This is a small amount of the extracellular matrix, your ECM, that surrounds muscle tissues, and it basically helps hold those muscle cells together. Now you're probably going, wait a second, I only mentioned striated and smooth muscle. There's 3 muscle types. Let's get a closer look here. Striated muscle can be skeletal muscle or cardiac muscle. And then of course, smooth muscle, and we'll talk about where these are found. Skeletal muscles, well, I mean, pretty obvious what they're attached to, right? Your skeleton. And they're used for movement. So every motion you make, every movement you make is a function of a muscle contracting. And if it's a movement for motion, it's a contracting mostly against bones. Now there's a couple of examples where it's not like your eye is blinking and things like that. But for basically when we move, your muscles are attached to a bone. So every motion you make, hand waving and everything, those are different muscles contracting and then relaxing. Okay. So one thing about skeletal muscle, it's voluntary, or at least most of the time. If you've ever had a cramp, that really sucks. Those muscles are contracting involuntarily, and that often is because of electrolyte imbalances and things like that. But almost all the time, it's voluntary. Or when you're shivering, that's another reason why they would be involuntary. And then the other thing is these muscle cells are long. Like if you get a bicep, they insert here and here. Well, an entire muscle cell will run the length of it. They're very long and very thin. And that sets up some interesting things like they are multinucleate. Multi, of course, means many. Nucleate means nucleus. So a muscle cell is multinucleate. It's got several nuclei in each muscle cell. That in itself is kind of interesting, isn't it? How does a cell end up with multiple nuclei in it? It turns out that when your cells are developing, you've got, of course, your myoblasts, of course, the blast being the immature cell. As they're growing, they merge together to form one long fiber, and that's how you get multiple nuclei in there. And then the reason why you have more than one nucleus inside of a muscle cell is because, well, your muscle cells are long. So you've got to control what's happening here, 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 in all these different parts of the nucleus. All these different parts of the cell. So if you start working out and you start building muscle, well, the information to make the actin and myosin and all the other proteins involved with your muscle are in your nucleus. But if your nucleus is down here, you're not going to be able to build muscle up here very well. So like I said, the nuclei are scattered throughout the entire length of the cell. Good? Makes sense, right? Okay. Yeah. So if I got everything I talk about there. You can also notice them because they have striations. You can see on this picture right here, of course, this is your nucleus. There's multiple nucleuses. These are the striations, and then this is the endomysium right there. And there's striations are caused by layers of actin and myosin that in some places overlap and would cause it to look dark. In other places, they don't overlap where it looks light. Okay. Any questions about muscle tissue? Yeah, it's pretty fascinating learning how the actin and myosin slide past each other. It's something I'm looking forward to teaching and talking about. The next one is a cardiac muscle. And the first thing you'll notice that it has is similar to the striated muscles, is that it's also a striated muscle, your skeletal muscle, is that it's got striations in it. I didn't say that very well. Sorry about that. Cardiac muscle is a striated muscle. You can see the striations in them. So that's immediately how we can see the similarities between a cardiac muscle and skeletal muscle. But there are some differences here. One, cardiac muscle is only found in one place in the entire body. It's pretty easy to know where, right? It's your heart. And first of all, whereas your skeletal muscles are voluntary, you make them contract. You control how much and how hard and how little they're going to contract to control all of your motions. Cardiac muscle is involuntary. You don't control it. It works independently of your brain, basically. I mean, it can respond to what you're doing. You start exercising, it'll speed up. You rest, it slows down. But it's involuntary. You don't have conscious control over it. Your skeletal muscle is a long single fiber. If you look at the picture, what you'll notice with the cardiac muscle, they're shorter and they're branched. So for those of you following it at home, we're on slide 5. So you can see that they're branched, and then they have these intercalated discs. And those are the dark lines separating the individual cardiac muscles. Skeletal muscles don't have those intercalated discs. And in those areas, they have gap junctions and modified tight junctions. Now you got gap junctions. What do you think those are doing in there? What does a gap junction allow cells to do? Well, what is a gap junction? You guys remember? Yes.
Does it allow space in between for like fluids to flow through?
Yeah, exactly. So what happens is you've got your cells are being stitched together by the tight junctions, right? And then you've got these gap junctions in there which form little pores going between the two cells. When you see lots of gap junctions in a place, that allows the cytoplasm to mix. It also allows for a signal to rapidly transmit through those gap junctions and allow these cells to communicate. That's also how your heart's able to beat as a whole, because these cells are communicating with each other. Okay. Also, the other thing is that heart muscles tend to be, like I said, shorter. They're bifurcated, and they only have one nucleus. Okay? Any questions here? The next one is your smooth muscle. This is another type of cell that is a muscle like your heart. It's involuntary, but it's not striated. Now, you have every hollow organ. Think like your esophagus. You know you can swallow laying down. You can swallow standing on your head. Astronauts can swallow. And the reason why is your esophagus has a smooth muscle that actually contracts and moves your food down to your stomach. And when you chew your food, you create around what's called a bolus that you swallow. And your esophagus can contract, making that happen. So blood vessels, eyes, skin, duct, we find smooth muscle all over the place. Also, a uterus is one giant smooth muscle. And once again, these cells are flattened. They only have one nucleus in the cell. You notice the cells are smaller, they're not as long. And the cells are linked together by gap junctions. That means those cells are communicating to each other adjacently. Okay? Yes.
So I have a question. Is there like a difference in the actual like makeup of I guess like usable muscle versus like strength muscle? Like is the muscle that like rock climbers have any like actually different than like what bodybuilders have, or is it just the way they train?
That is a really good question, and I was going to get into that when we start talking about muscles. But you do have different muscle types. You've got type 1 and type 2. So you have like a fast oxidative glycolytic muscle and a slow oxidative glycolytic muscle. And then you have, oh, what's the other one? The slow twitch. I forgot what it's called right off the top of my head. So a rock climber and a bodybuilder are actually going to have kind of similar muscles because of the strength involved with that. Whereas, like, if you compared Arnold Schwarzenegger to Lance Armstrong, is it okay to still talk about Lance? I know he cheated, but the dude, like, it doesn't matter. It's rampant in the sport, and it doesn't matter. He still, like, is an impressive human being in terms of his cardiac output, his endurance output, and cardiac ability. Doping or not, he's still amazing, like, as an athlete. Same with a lot of your bodybuilders. They're still athletes. The difference in those is in those types of muscles, like a fast twitch muscle, when I say glycolytic, is burning carbohydrates. So it's more of an anaerobic, doesn't have a lot of mitochondria in there, and they're capable of exerting a lot of force rapidly. That's why bodybuilders and weightlifters can lift a lot of weight. But you put us on a bike and we suffer. On the flip side, your slow oxidative muscles, that's what it's called. Your slow oxidative are relying on aerobic respiration, so they're much denser with mitochondria. They have a lot more myoglobin, which makes them look dark, versus a fast twitch, which looks white. So think like chicken, white meat. Where's that found? It's breast meat. Chickens can fly very fast for a quick burst, but they run around all the time. Their dark meat is fuller of myoglobin and mitochondria, which gives it a dark. But they can't generate a lot of force, but they have a lot of endurance. So yeah, there's definitely trade-offs on these type 1 and type 2 muscles. And I was going to catch up on my research on that to know whether or not they can switch, or if you're born with a certain amount at birth or not. Yeah. Any other questions? Yeah, we'll get into that more when we talk about the actual muscles, because I was going to do a little bit of extra research on the slow twitch, fast twitch muscles, and we'll see what the latest findings were. Okay, let's look at nervous tissue. Nervous tissue makes up the majority of your brain, spinal cord, and of course your nerves. And it's composed of two main cell types. There's lots of different types of cells, and their surrounding extracellular matrix. So of course the neurons. These are your excitable cells, and then you have the neuroglial cells. The ECM is also unique. It's made up of a ground substance with proteoglycans that are unique, and you don't find those specific proteoglycans in other places. And there's also very few protein fibers. So when we start talking about nervous tissue, the neuron, that's the primary cell type. And this is the one that can receive and send signals. And like I said, it does it right down the axon, which is the long part of the cell. Okay, then there are these neuroglial cells. And there's a fair number of them that we'll talk about. And they have different functions that are involved with support of the neurons. And it turns out that, like, some of our latest findings in how our brain works and how we get things like dementia, Alzheimer's disease, Parkinson's disease, we're learning that these neuroglial cells are way more important than we thought. We thought they were just there to support these neurons, and it turns out that they're quite important in the functioning of our brains. And when they go bad, things happen. Yeah.
Isn't it true that we are born with a certain amount of neurons and neuroglial cells and they don't regenerate themselves?
No, not at all, actually. I hate to start to shoot you down like that. It's interesting. Like my daughters, right? Their brains are growing like rapidly, adding cells all the time. And what's going to happen is, I gotta beef up on my exact timelines here, but basically, when their head gets about the size of an adult, tweens, their brain cells will stop growing and dividing at that point. And so you can see like the baby's head grows. They're adding on lots, billions of neurons and glial cells and things like that. Then in teenage years, things start to happen. You start losing brain cells as a teenager. No, doesn't make teenagers any dumber. We think so, but not really. What happens is, as we have all of these extra neurons, a lot of them aren't needed. So the ones that are actually forming connections and have purpose will stick around. The ones that aren't being used, you're literally like you're thinning out the herd. And by doing that, you actually speed up your brain. You make it more efficient. Good? Yeah. But after that, it becomes pretty fixed. And same with your muscle tissue. It becomes fairly fixed after a certain age. And like I said, I forget all of those developmental milestones. I haven't kept up with them, but I'll look them up for when we start talking about these. Good question, though. Anybody else? Yeah. I love it when you guys ask questions. It's fun to talk about these things. So a neuron, like a muscle cell, basically once they reach adulthood or they mature, they no longer replicate. They can, but they don't often do it. And one of the things that we've been working on is trying to like stimulate our nerve cells to reproduce. And you can imagine if you have nerve damage, you want to be able to reproduce those nerve cells so you can fix your nerve damage. The problem is getting those nerve cells to grow back into the right place again. Like if you have a severed spine and you've lost control of your legs, it's how do you get those nerve cells to reconnect to those muscle cells and coordinate those muscle cells, and that's proving pretty tough. Okay. So the thing about neurons, I've said this once, they're excitable cells. Once mature, they typically don't divide by mitosis. There are exceptions to that, of course. They have the main part of their body. So when you see the middle part, that's your soma. And then that's where the nucleus is. And they also have these things called the dendrites. Hard to see it here, but these are the dendrites coming out. So this is the soma. Anytime you see the word soma, think body. Then we have the dendrites here coming out. And those are what send out from the main nerve cell and allow for communication to occur. Okay. So we have these neuroglial cells, and they have a lot of functions. One of them is they act as anchors. They hold things in place. And things they hold in place are like the neurons, of course, and also all the different blood cells that are, or the blood vessels that are, the blood vessels in your brain. They'll hold those in place. They also monitor the extracellular fluid, part of this ECM here, and make sure it maintains what it needs to be. They can also, in some cases, they can speed up the rate at which an impulse is sent down the axon. Okay, and we'll learn about those. Those are called Schwann cells in the nodes of Ranvier, where you can actually have the nerve signal go very fast because of that. And they also help circulate fluid around the brain and the spinal cord. If you know there's fluid in both, to help protect it, and they help circulate that fluid. And the neuroglial cells, they can reproduce by mitosis. Okay, let's go on here. Any questions? All right. So this is 4-6 here. We're putting it all together. So you get the idea. We have approximately 200 cell types in our body. And cells that are similar in what they do in their function form tissues. And then if you get 2 or more different tissues, you start forming organs. And then organs, of course, form organ systems, and that makes us who we are. Okay. So a simple organ, skeletal muscle, it's got the skeletal muscle and dense irregular connective tissue, and each of these have a different role, right? So the skeletal muscle, you know what that's doing, it's just allowing you to contract. And then the connective tissue, that's what holds us together, all the collagen. And together, that forms an organ. It was interesting reading the textbook and reading about like the trachea. You know, it's something that's, you know, it's right here. You know, we use it for all kinds of things here, swallowing, air passage, you name it. And I never realized like how complicated something like the trachea was. It's easy to say, you know, the brain is complicated, nerves are complicated, but the trachea is this hollow organ. I mean, it allows for air to get in and out of your lungs, right? But it's got different layers of tissues to it. So it's got anterior, posterior end. It's got dense irregular connective tissue on the top end up there. It also has smooth muscle. So if it has smooth muscle, you know it's contracting. It's got hyaline cartilage in it. It's got loose connective tissue and pseudostratified columnar cells that are ciliated that beat back and forth to help keep stuff out of your lungs or get stuff out of your lungs. So something simple like a trachea actually has more tissues and parts to it than like a muscle cell, or I'm sorry, muscle tissue. Okay. Any questions? All right. Let's talk about membranes. Yeah, you know, I grew up in the 80s and the 90s, and every time I hear the word membrane, you know, Cypress Hill, I'm insane in the membrane. That song does not get out of my head ever. It's been up there for like ever. What a great song. I introduced it to my daughter, so she'll start going around singing it too. Okay, true membranes. They have a superficial epithelial layer, and this rests on top of a connective tissue layer. And sometimes that might be like it might contain some smooth muscle in there as well. And we'll get some examples of this in the integument. So these are thin sheets, the membranes, and they line a body surface or a cavity. And then we have true membranes. These are the serous membranes and the synovial membranes, or synovial. And we also have membrane-like structures. And these include like mucous membranes as well. Okay. Let's start with a true membrane. And if you look up there, you see it's got a picture of the lungs and the trachea coming down into the lungs. And these true membranes, they don't open to the outside of the body. And of course, these are the serous membranes and the synovial membranes as well. And serous membranes, they fold over themselves and look like they have 2 layers. So the outer layer is, of course, the parietal layer, and that lines the body wall. The inner is a visceral layer, and this covers the organs inside the body cavity. Well, when the synovial membranes get something wrong with them, you hurt in your joints. You know, your joints, if you think about it, every joint around here, you've got bones, you've got your connective tissue holding it all together, and then you've got muscles that allow you to contract. And one of the problems is that as we age, we lose some of the connective tissue in there, and that can start to hurt when you rub bone on bone. But also around that joint is you have this synovial membrane. And that synovial membrane, on the inside of it, you've got these, what they call synoviocytes, and you know, and some loose connective tissue, and then some dense connective tissue. And they release a fluid. So your synovial membrane maintains this like slippery fluid around all of your joints that allow them to like keep moving easily. They just kind of lubricate it. So the problem with like when you say one of my synovial joints is hurting, your joint is going to hurt if you have a problem with that. Okay. And then, like I said, so you've got these two layers. The outer layer is basically, it's a loose and dense irregular connective tissue, so it makes it strong. Then the inner layers, you've got your modified fibroblast. You see that word blast again. That tells you it's an immature cell that is doing something. And that secretes your synovial fluid. And that fluid, like I said, is very slippery. And that provides lubrication to your joints. So bad things happen when synovial joints go bad. Okay. Next are membrane-like structures. This one, you've got the cutaneous membranes and the mucous membranes. They don't quite fit the description of a true membrane, but they do line all the body passages. And because they open up to the outside, that's why they're not a true membrane. They're membrane-like. So they're in your respiratory passages, they're in your mouth, your nasal cavity, your digestive tract, and in both reproductive tracts for men and women. So here they go. They've got an epithelium and its basement layers. And then the layer of the connective tissue is called the lamina propria. And it's got a thin layer of smooth muscle around it. And it's also got goblet cells. When you see goblet cells, remember what they do? Other than make us think of the goblet of fire or something like that. What do goblet cells secrete? Mucus, right? So if you think about it, we have mucus that lines our stomach. Why would we have mucus lining our stomach?
Maybe access protection.
Absolutely. It protects your stomach from pathogens, and it also protects it from your own digestive enzymes and the hydrochloric acid. Your stomach is really acidic and got some pretty good enzymes in there to break things up. And in fact, when people get ulcers, part of it is that mucus lining gets compromised and you get Helicobacter in there. You get a bacteria that starts eating at the lining of your stomach. Okay, so you've got all these mucus, and that mucus, it lines all of these, tracts that open up to the outside world to help protect them, including, like I said, your stomach. Okay. Any questions? Okay. The other one is your skin. It's called the cutaneous membrane. And of course, the skin, you probably have heard this before, but it's the largest organ in your body. It weighs like 15 pounds or something. It's big. You get the outer layers. And if you remember, we went through our tissues, right? And you remember the 3 different cell types? Squamous, cuboidal, columnar. And then on your skin, you've got keratinized, stratified squamous of the epithelium. That tells you a lot. I know it's a lot to remember. What does keratinized mean? Yeah, it's got dead cells, but it also refers to they get a lot of keratin in there. And keratin is an intermediate filament. It's a permanent structure of your cytoskeleton made of very tough proteins. That's what makes your outer skin so tough. It's also it's protection too, right? Then you have your dermis. This is a loose connective tissue. It's found beneath the epidermis. And then there's an even deeper layer of dense, irregular connective tissue. And in there, you can get like blood vessels that provide oxygen to the other cells and allow also nutrients to diffuse upward as well. Okay, any questions here? Yeah, so when you scrape yourself, if you start bleeding, you've basically ripped off all of the keratinized stratified squamous epithelium, and you've gone all the way down into the dermis, and that's where you started busting those blood vessels. Okay. Yes.
It kind of goes back. I just have a question. Like, what's the difference between your bursa and like the, I don't know how to say this.
Synovial?
Yeah.
I'll get to get back to you on that one. Sorry.
Is it roughly the same? Like,
They're similar, yeah. I gotta look it up before I give you an answer on that. I'm not gonna try to bullshit an answer here. Sorry. If this was an evolution question, I could probably get my way through it, but it's got a specific definition, and I just don't remember it right this minute. Like I said, it's yeah. Good question though. I'll remember to look that one up and give you guys an answer. Okay, now we're going to move on to tissue repair, and let's talk about regeneration versus fibrosis. So we've all had cuts, scrapes, scratches. Hopefully nobody's had anything worse, maybe a broken bone here, or if you're like me, I got a torn pectoral muscle, awful. We all get injured at some point of varying degrees, and the tissue repair is wound healing. And so you can remove dead and damaged cells, and in some cases you can replace those cells with living cells, so you have a complete repair. And then other times you can't. You get what's called fibrosis. And so while some tissues can regenerate, where the dead and damaged cells are replaced with the same type of cell, others not so lucky here. On your skin, it makes sense, right? Because we're exposed to everything. I mean, every time I go on a hike, there's a new scrape or something, it seems like. And so basically your skin, not only is it regenerating cells all the time, but if you scrape yourself, it can repair, it can regenerate new cells. And if you don't have a bad cut, you can't even tell you're ever damaged. And for those of you interested in cell biology, there's some really cool stuff about like how skin cells grow. And if you've ever noticed, like our skin is nice and smooth, right? It's not growing all over the place. I mean, every now and then people might get a skin tag where it grows up. But when skin cells, whenever you scrape yourself, the skin cells will realize there's nobody over here, right? So if you're all bunched up together, the skin cells are going to be growing from the bottom, and then they'll die and slough off. But if you damage them, all of a sudden there's not a live skin cell right there. That skin cell says, wait, there's nobody there. And that causes a signaling for that cell to start enter into the cell cycle and start dividing through mitosis to fill in that gap. And then when the skin cells all grow back together again, and they're all touching, then they'll stop. And that's how like our skin doesn't just keep growing all over the place. It's pretty interesting. When it goes wrong, people have real problems. Okay. Sometimes our tissues, they basically are not able to regenerate. So we get fibroblasts. Once again, there's that word blast. It's like an immature cell. And these fill in the gaps by a process called fibrosis. And this is where you get scar tissue from. Of course, every time I hear scar tissue, I think of Red Hot Chili Peppers, right? And scar tissue, what they're doing is these fibroblasts, they're dividing by mitosis, and then they're making collagen to fill in those gaps. So the problem with that is you can fill the gaps, but you're filling it with fibers, which is not what the original tissue was. So then that's how you get scar tissue, and you don't have the same level of functional ability. So when you really damage like your skin and badly all the way down to the dermis, if you damage it bad enough, it can't grow back, and you start just filling it with scar tissue, and this is fibrosis. So it was 2 years ago, I fell off my deck and got caught on a nail and ripped this whole area right there. And I I have a nice scar to remind myself to remember to remove my nails so I don't leave them out hanging on my deck when I fall off of it. I was working on it. Okay. So yeah, scar tissue, and that scar tissue, think about this. It's dense, irregular connective tissue. Dense, irregular. So if you have scar tissue like in your muscle, it's not going to be as elastic. Okay. And yeah, so basically scar tissue doesn't function quite the same. So whether or not you have tissue repaired or whether or not you get scar tissue is basically it's determined by that cell's ability to regenerate itself, to undergo mitosis. So, you know, you lose 50 billion cells a day, right? 50 billion. That's part of our normal process. And we lose those in our epithelial cells. So your skin, your esophagus, your stomach. So those areas, if they get damaged, they are actually pretty capable of regenerating themselves and fixing most wounds. And they do this from these things, these stem cells. You guys know what stem cells are, right? There's different types of stem cells, but stem cells are kind of undifferentiated. They're not mature. And as they divide, they'll receive chemical signals from their environment, and then they'll begin to differentiate where they start to express all the proteins involved that make them into a skin cell or a stomach cell or whatever. Let's see. So some cells, like liver cells and blood vessels, they actually, if they become damaged as mature cells, they can divide, but only when they need to, whereas skin cells and epithelial cells are dividing all the time. In bone, believe it or not, connective tissue proper that includes bone, and they can actually regenerate as well through these immature cells. So most connective tissue can regenerate, except for cartilage, which I wonder from an evolutionary point of view why that's the case. But cartilage is the exception. They have these resident cells, but those cells just have a limited capacity for regeneration. So you get a lot of fibrosis whenever you damage your connective tissue. Man, I put to get like 10 people to sleep today. Wow. All right. Doing good. I know. So if you think about, if you tear your cartilage, and if you remember like your cartilage, like that forms a tendon or a ligament, it's not dense or regular, right? It's smooth and stacked. And what happens is if you tear like a tendon or your cartilage that's holding your joints together or your muscles together, if you tear that and then it grows back, there's a lot of fibrosis in there. You lose mobility if you have a lot of scar tissue. Okay, so, and also it just, man, if you damage your connective tissue, it just, your tendons and ligaments, it takes forever. Interestingly, smooth muscle can regenerate, but cardiac and skeletal muscle generally heal by fibrosis. So like a few years ago when I turned 40, imagine that you turn 40 and the first thing I do is like tear my pectoral muscle. I'd been working out for, gosh, since I was 15, so like 25 years. Never had a muscle tear. Turned 40, bam, muscle tear. That basically ended my competitive weightlifting days permanently because because when it starts to heal, it's not going to heal back quite the same. It's going to have some scarring in there. A mature skeletal or cardiac muscles also, they cannot regenerate themselves. You can imagine that people with heart disease, this is a real problem, right? Whereas you don't need all of your pectoral muscle to function on a day-to-day basis. I still function just fine. When people have heart attacks, what's happening is that sometimes part of that heart might actually die and it doesn't regenerate. So what happens is as you accumulate damage to your heart, its ability to contract and move blood throughout your body becomes diminished, which puts stress on the rest of it, right? So that's one of the problems. So you can imagine that like an area of research that people are trying to do is figure out how we can get your heart to regenerate itself. How do you set the clock back? Because at one time, as you had this really good question, like, about our brain cells, are we born with a set amount? And the answer is once we reach adulthood, we have basically a set amount, right? Same with our cardiac muscle, same with our skeletal muscle. But at some point, they actually grow. And the idea is how can we reset these tissues to a younger age and have them grow and replace the damaged tissue? So that's something that scientists are trying to figure out how to do, right? How we can switch these cells back into becoming mitotic again, get them to divide. Good? Yeah, it's not too hard to get them to divide. It's just to get them to grow correctly is another problem. Okay. Yes?
Can you say about satellite cells?
Not so much. Not right now. Like I said, we're going to get into more detail on these things later. This is just kind of introducing here. Forgot my water. My throat's starting to give out on me. Okay, so capacity for tissue repair. Nervous tissue, these nerve cells generally do not undergo mitosis once they reach a maturity. So what happens is if you have nerve damage, you get fibrosis. However, neuroglial cells, they can still divide by mitosis and replace dead and damaged neurons with scar tissue. So if you have nerves or neurons that are getting damaged, your glial cells are what's going to fix that tissue as best it can. And interestingly, outside of your brain, when you start getting into the central nervous system, once you get out of that, and you get out, the CNS is called your central nervous system. Once you get out of that, like nerves in the rest of your body, if the axon becomes damaged, as long as the body part of it, the soma just means body, if it's still alive, it can actually regrow axons. So sometimes people with nerve damage do actually gain function back again. My finger right here. I'm actually getting some function back after a couple of years. I was cleaning my fish tank and I had a sea urchin spine, which I didn't realize they were venomous. Punctured and went all the way to the tip of my finger. That's why I didn't go all the way through. It hit my fingernail, the tip, and it delayed me going on a trip for a couple of days. And I had to walk around like this because it hurt so bad. But for the longest time, this tip of my finger was completely numb for like a year. It's now about 50% numb. You never think about how much you need to use the tip of your finger for stuff. Good thing my nerve cells regenerated, right? Okay. There are a lot of factors that affect how well a tissue can repair. Nutrition, blood supply. These really, really matter. If you do not have a lot of blood supply to an area, you can't get enough nutrients to there and remove waste. So those cells, they're not going to grow very fast at all. And interestingly, you guys know that we need to have vitamin C, right? I mean, that's like something we have to have that we get from fruits and vegetables. And do you know what happens if you don't get vitamin C in your diet? Anybody know? Yeah, you're right. You get scurvy. So interestingly, before we knew, so several hundred years ago, you'd get these boats, these ships, and they'd go out and these sailors would sail around the world exploring. And one of the biggest problems they faced is They could pack a lot of food on there onto the ships, but things like fresh fruits and vegetables didn't last very long, right? Like maybe a couple weeks at best. So if you were going on a long voyage, they would run out of vitamin C. They would run out of fresh fruits and vegetables, and the sailors would start facing scurvy. And it starts with like you bruise really easily. You start bleeding from your gums. It's awful. It's nasty. And the reason why you get scurvy from not having vitamin C is because to make collagen, your cells require vitamin C. Pretty wild, isn't it? Now that should beg another question from an evolutionary point of view. And that is, why in the world do we not make vitamin C? You know who does make vitamin C? Does your dog need to eat fruits and vegetables? Does your cat need to eat fruits and vegetables? We do, right? It turns out that most animals that can make vitamin C, they have a series of genes that they have the metabolic ability to make vitamin C. So your your pet makes vitamin C. They don't need it in their diet. However, the ancestors to humans millions of years ago switched from a diet to include fruits and vegetables. And we think that's one of the reasons why we see such good color. We see more colors than like a dog does, because we had another mutation that led to more different types of proteins in our eyes to see color. So we have a pretty wide array of color, so we could pick out fruits and vegetables. So as we developed better eyesight and had a more varied diet, we got plenty of it from of vitamin C from the diet our ancestors had. So eventually the genes to make vitamin C mutated and no longer functioned in our ancestors. And that's okay, because we were getting plenty of vitamin C from our diet. So we actually have a pseudo gene. It's a false gene, but it's a remnant of a gene to make vitamin C. Each one of us carries that. Isn't that pretty wild? It's there, it just doesn't work. Yeah.
Okay, I have a question. So for a disease like scleroderma,
Okay.
Since it's tissue repair, is it speeding up the repair process?
Well, scleroderma, yeah, that's where you're getting like all the huge, sclero means like scaly, right? Yeah, so obviously there's something wrong where you're building up too much of that, the keratinized layers. Yeah, so there's something wrong where the cells are dividing too much and they're not stopping when they need to. And I think that's the issue with that.
And that's what causes it to be like stiff?
Yeah, because it's filled up with keratin and it's getting dried out. Yeah, I think that's fairly rare, fortunately.
Yeah, my aunt has it, so.
Yeah. Okay. All right. Any other questions? Yes.
What happens to our skin when we get stretch marks? Like, why is the skin not repaired and why does it stay visible?
Yeah, so I've never really known like how stretched. I know that you get them because the skin is getting stretched too much, and I think you're just creating separation between the outer layer and the dermis, and it's not filling in correctly, so you're getting some fibrosis in there. Any other questions? Okay, I'm gonna hold off on starting on chapter 5. I forgot to drink water, so my lectures to give, and I need to get some water in a bad way right now. So I'm going to stop here, but really quickly, pay attention to school closings on Thursday. We have a really bad cold front that is headed our way, and it's going to start snowing. And for those of you that know, I do live way out east of the mountains, and I have to drive through a pass. And if I wake up and there's a foot of snow on the ground, I'm probably not going to drive into school. Also, school might be closed anyway, so keep an eye out for school closures. If I do come in and you don't feel safe coming in, that's fine. But definitely check your email to see if I have class. If we're on a delay or anything like that, I'll just do it from home. Look for the live link. I'll send out an email. If I go live, I'll do it from home. And we'll start chapter 5 on Thursday, whether we're in class or I'm at home just live streaming it. Okay? All right, you guys have a good one.
So I actually had 2 questions.
Okay.
One, when you're connected